Okay, so this lecture is going to focus on the properties of cells, what is contained inside all cells, what are some key differences between prokaryote cells that are like bacteria and eukaryote cells that are like the cells we find in our bodies. In particular, we're going to focus on eukaryote cells and the organelles or compartments that are found inside eukaryote cells that allow each organelle to perform various different jobs for the cell. We're also going to briefly talk about what mitochondria and chloroplasts are, why we do not consider these to be organelles, but rather the remnants of once free-living prokaryotes. Okay, the cell is basically our smallest measurable unit of life, essentially. We do have particles, of course, called viruses that we're talking about in class right now that are much smaller than cells. But viruses are technically not alive because they have to have a cell in order to replicate, right? If they're outside, they're just basically inert molecules. So a cell is basically our simplest, smallest measurable form of life. And as we've seen with everything so far in this course, the structure of a molecule very, very closely matches its function. So we can see, for example, in the middle here, we've got a white blood cell, which is this big blob here. Much larger than our red blood cells. Red blood cells, of course, predominant function is carrying oxygen to different parts of our body. And the form of the red blood cell takes is like the form of hemoglobin, which is the molecule that's carrying the oxygen that resides inside the red blood cells. So likewise, we also have a neuron here on the right-hand side. So here I've got the nucleus and all the other parts of the central body of the cell, but all these long projections, right, are all connected to other neurons or to sensory cells that are relaying information, and we're going to talk about that more at the end of the course. But so that long spindly form means that it can connect to different types of cells and neurons elsewhere. Likewise, also we have a kidney cell here on the left, or what's called the nephron. And it's basically a long series of kind of long tube-like cell. The cell body is actually, elsewhere. But it's essentially these long tubes, and there's a lot of diffusion that's happening between the bloodstream and the nephron, and between the nephron and this collecting duct here that then goes to your bladder. So the form the cell takes is directly related to the function that it performs. Okay, so basically what makes up a cell, whether it's a prokaryote cell or a eukaryote cell? All cells contain, or are bound by, rather, a plasma membrane, right? This is predominantly made of the phospholipids that we were talking about earlier. I believe last week we were talking about lipids, where we have the hydrocarbon tails that are in, or the fatty acid tails that are in the center, and they're attracted to each other because they don't want to have anything to do with water. And then you have the phosphate heads that are very hydrophilic, are facing out either into the center of the cell or into the external environment. For all cells, we also have in that membrane embedded proteins. And these proteins act essentially as transport mechanisms. So they allow ions to cross the membrane because it otherwise would not be able to do so. And it allows other kinds of molecules to be transported into and out of the cell. And when we get to transport next week, we're going to spend a lot of time talking about that. They also, of course, contains inside the cell a semi-fluid substance called the cytosol. This includes basically a lot of floating molecules like sugars and proteins and other stuff. Excuse me. You may have also been able to see possibly in lab this week what is called cytosolic streaming. So it's the movement of that fluid inside a cell as it moves around different concentrations inside the cell. You may have been able to see that with the amoebas that you were looking at under the microscope. All cells contain molecules of inheritance. We use DNA. So all cells use DNA as their genetic material. And these are composed of long thread-like molecules, either in a circular loop or basically as long strings, which is what eukaryotes use. And that, of course, is containing our genes. So we refer to them as chromosomes. A chromosome is essentially a long strand of DNA that contains, genetic information. And finally, all cells have ribosomes. Ribosomes are made up of protein and what's referred to as ribosomal RNA, a molecule. So it's a specific type of RNA that's only found in ribosomes. And those two things together work to read the messenger RNA instructions that are coming from your DNA template as to how to build proteins. And so these are our protein assembly structures that you find in all cells. So all cells are going to have genetic material, chromosomes. All cells are going to have ribosomes. All cells have this fluid that basically carries everything the cell needs, right? The sugars, any other molecules that are floating around in there. And it's all bound by a plasma membrane. Okay, so don't worry about memorizing this table, but mostly this is to help illustrate the differences that we see between prokaryote and eukaryote cells. So in general, we're going to focus on the features in the red boxes, right? So major difference between prokaryote cells, right, which includes things like bacteria, and eukaryote cells, which includes pretty much everything else that we are aware of on the planet. We have a defined nucleus in eukaryotes, and it's something that is bound by a layer of membrane that we call the nuclear membrane. Prokaryote cells have their genetic material is just sort of in a region that's kind of packed together in part of the cell, but it's not bound by a membrane. It's not in a sort of a separate compartment. And so that's a very consistent and important difference between prokaryotes and eukaryotes. How our DNA is structured. As I mentioned earlier, eukaryotes, we have linear chromosomes. So basically they're like long strings, whereas bacteria and other prokaryotes have their DNA is arranged in a circle. It's basically a loop. And there's no beginning or end, to it. They also may have some other smaller loops that we call plasmids, and we'll be talking about that more in lab later on when you guys do some experiments with those. Another major difference between prokaryotes and eukaryotes is the presence of membrane-bound organelles. So these, again, are basically cellular compartments that are surrounded by membrane. It's going to be plasma membrane, much like the cell membrane, but the proteins embedded in them are different based on the function of the particular organelle, and that's going to be a big part of the focus of this particular lecture, is talking about the jobs that are performed by those different organelles. Bacterial cells and other prokaryote cells do not have membrane-bound organelles. Consequently, as we can see in the bottom of the page, prokaryote cells are a lot smaller than eukaryote cells, right? Because eukaryote cells have a bunch of other stuff in them, right? They've got a membrane-bound nucleus and they have all these organelles, and those are things that take up space. And so because prokaryote cells don't have any of those features, they tend to be quite a bit smaller than eukaryote cells. Prokaryote cells also have a very clearly defined cell wall that's outside of the plasma membrane, and that's an additional layer of protection and support for them. These are single-celled organisms that live in a variety of different kinds of environments, and that cell wall basically helps protect them from their environment a little bit more, keeps them from drying out, and for those who live inside the tissues of other organisms may actually help protect the bacteria from the host environment. Eukaryote cells, by contrast, some eukaryotes have a cell wall. It tends to be made of a variety of different materials. So fungi have a cell wall that's made of chitin, very much like the shells of arthropods that we were looking at earlier when we talked about um, um, structural sugars. Plants have a cell wall that is filled with cellulose, and that's how they support their cells against gravity. Other eukaryotes have cell walls that are made of silica, and so they end up looking like little grains of sand, even though they're alive. Those would be diatoms. So we have lots of different things that you could potentially have a cell wall that's made of. Animals actually don't have a cell wall. So our cells don't have an extra boundary around the outside. Our cells are held together instead by connective tissue and including collagen. And you'll notice the common features for both prokaryotes and eukaryotes, right, are those features we just talked about earlier, the plasma membrane, the ribosomes, and just a note about the ribosomes, right? Ribosomes in prokaryotes serve the same function, and basically do the same basic job, and they're also made of protein and ribosomal RNA, but the structure of ribosomes in prokaryotes is different and distinct from the structure of ribosomes in eukaryotes. And that helps explain why antibiotics, for example, which are chemicals that inhibit the growth of prokaryote cells, in particular bacteria, why antibiotics affect bacterial cells and not eukaryote cells. Okay, so here's just sort of a close-up zoom of a typical prokaryote cell or bacterial cell. And we've got the scanning electron microscope image on the right here, and then basically a diagram on the left. And so you can see the chromosomes are, or the chromosome singular, excuse me, basically a very, very, very long loop of DNA that's just sort of contained in a particular region. There's no membrane that surrounds that. All these little dots, right, are the ribosomes, and those are the sites of protein synthesis in both prokaryotes and eukaryotes. We have a flagella, which we're going to get back to when we talk about eukaryotes some more. And that's basically just a long filament that is attached to mechanisms in the membrane and the cell wall that allows the bacteria to move through its environment. And we also will notice, right, we've got a plasma membrane. Yeah, there we go. It's that kind of pinkish bit here. And then you have the cell wall that's outside of that. And that, of course, is where our flagella is anchored. And then bacteria have an additional layer on the outside called the capsule, which in this case is very well defined, so we call it the capsule. If it's kind of mushy, we call it a slime layer. I gotta love these terms. Okay. And the only reason that's important is that capsule layer is just an additional layer of protection. It helps bacteria stick to a surface. It helps them move. It keeps them from getting dehydrated, and can help protect it from a host immune system in the case of bacteria that live inside the tissues of other organisms. So the things you're not going to see when we look at eukaryotes here next are the cell wall and the capsule. The nucleoid instead will be a nucleus that is bound by a layer of membrane. Okay, so you're probably thinking, ay, 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 what am I supposed to do with this, right? So this is kind of a make-believe eukaryote cell. I can't think of any one single cell that has every single one of these features, in particular the cytoskeleton features like the flagella and microvilli and some of these other things. However, most eukaryote cells have certain things in common, right? So they have the nuclear the nucleus, right, which is our region with the DNA, and then you have the nuclear envelope around the outside. You obviously have the plasma membrane, right? That's your outer boundary of your cell with different proteins and other things embedded within it that allow the cell to move different kinds of molecules and ions into and out of the cell. They have what's called the endoplasmic reticulum. We have two different kinds, a smooth and a rough. And we're going to talk about the different jobs of the endoplasmic reticulum and why it's generally found kind of wrapped around the nucleus, although there's lots of little gaps here. You have the Golgi. This is kind of our shipping and receiving center. The mitochondrion. You often do have more than one. Some cells have a lot of mitochondria. Some only have a few. This is an animal cell because we can see that there's no external cell wall present, and there's also no chloroplasts present. Those are the sites of photosynthesis, and we're going to be spending more time later on talking about those. They did include the centrosome and the microtubules in here. These are part of the internal skeletal system of the cell, which we sort of think of like cells have skeletons. Yes, they do. It's microtubules and microfilaments in particular are providing a lot of structural support. They allow essentially work like a network of filaments that connect one part of the cell to the other and allow for movement of different types of molecules and around the cell, essentially allow the cell to talk to itself and send things from one organelle to another, or send it outside the cell or bring it into the cell, giving it, again, some more structural support. And in particular, the microtubules become really, really important when we start talking about cell division and mitosis. And so we're going to get to that more later. We're actually going to spend some class time going over the cytoskeleton, so I won't be touching on that anymore today. And we can also see we have a couple other of our smaller organelles. Here's what's called a peroxisome. So it's, again, a membrane-bound organelle. And we call it a peroxisome because it's actually filled with hydrogen peroxide, which is a pretty caustic chemical for those who know. You have the lysosome. We have a few of those as well. And as the name implies, that's where things get lysed. They get cut apart into pieces. So this is, again, kind of your everything but the kitchen sink sort of eukaryote cell. And we're going to go through each of these parts, though, in turn. And as I mentioned, the cytoskeleton, we'll cover that during class time. Okay, so our nucleus, right? This is where we contain our genetic data for animals, right? And eukaryotes, this means that you're dealing with linear segments of DNA that include all of the genes that code for all of our characteristics and traits. It's bound by actually a double plasma membrane. So it actually has two layers of membrane that constitute the nuclear envelope. And the reason for that is additional layers of protection and to regulate what can go in and access your DNA, right? Remember, this is your original code for a cell, right? You don't want anything to damage your DNA. Those are the instructions. If your DNA gets damaged, it's not going to function properly, and you may have actual changes to how the cell works. And for example, in a lot of cases, damage to genes that control the rate of cell division contribute to the occurrence of cancer. So you really definitely want to make sure you're protecting your DNA in there and not allowing just anything to access it willy-nilly because it may get damaged. So you have extra layers of protection around that. Having said that, right, it does need to talk to the rest of the cell, right? Molecules that are specifically formulated to modify genetic material, and we're going to talk about that more later on, that change how our genes are expressed and regulate that, need to be able to get into the nucleus. There are certain cell components that are actually made in the nucleus, and that is the I just said nucleus, excuse me, nucleus. There's no extra U in there. Nucleus. And these are, for example, ribosomal RNA, which is a fundamental component of ribosomes. Now, ribosomes are found outside the nucleus, and the protein for the ribosomes is synthesized outside the nucleus. But the RNA strands that function as the enzyme in our ribosomes, those are synthesized inside the nucleus, and so they have to be able to get out. Likewise, when we read our DNA and make a functional copy that we refer to as messenger RNA, those molecules need to get out of the nucleus and to a ribosome so that they can then be translated into protein. And I think I just mentioned this. Yes, yeah. So our ribosomal RNA is synthesized in the nucleus and then shipped outside to interact with proteins to form the ribosomes. So we do not find ribosomes in the nucleus, only outside of the nucleus, but the ribosomal RNA needs to be synthesized inside the nucleus. Okay, so here's just looking at, again, a kind of a close-up of our nucleus. So this is the image that we saw in the earlier cell diagram. Here's an image of the pore complexes, so you can see the ribosomes outside the cell, outside the nucleus, and then you have these little pores that in the nuclear envelope that are allowing certain things to come into the nucleus and other things to be exported outside of the nucleus. And that's pretty much the practical upshot of this particular slide. You'll also notice, right, that's contiguous with the nuclear membrane, the nuclear envelope, is the rough endoplasmic reticulum. And the rough ER, its main job is making protein. So if you are, in the nucleus and you're sending out working copies of your DNA in the form of messenger RNA, those are the instructions on how to make protein. Those can immediately dock with ribosomes right outside the nucleus and start that job. They don't have to go very far, really, in order to hook up with our protein synthesis centers, which are the ribosomes. Okay, so we have two major types of ribosomes, and the real difference is where they're located and the kinds of proteins that they synthesize, that they make. So what we call free ribosomes are ones that are basically floating in the cytosol, or the cytoplasm, and so they tend to make proteins that are needed to function in the cytoplasm. They don't necessarily need to be shipped anywhere. They're not proteins that will be used in other kinds of organelles. Bound ribosomes are ones that are basically attached to the surface of either the nuclear membrane, like we saw earlier, or they're attached to the surface of the rough ER. And those bound ribosomes are going to synthesize proteins that need to be used in membranes that are part of organelles or the plasma membrane of the cell. They also make proteins that need to be used in specific organelles, or proteins that need to actually be shipped outside of the cell. So ribosomes are not part of the endomembrane system because they're not bound by membrane, but they certainly make proteins that are used as part of the endomembrane system. So basically, if you think about all of our nucleus, right, that's bound by two layers of plasma membrane, we have our outer cell membrane, and then you have the membrane-bound organelles. Obviously, we've got a theme here, right? And they're all connected by this, by the fact that they are bound by the lipid bilayers. And what's cool is you can have the lipid bilayer basically siphon off a little section, and it ends up with basically a membrane-bound bubble. And membrane-bound bubbles can then also merge with other membranes and sort of become part of the whole thing. And so our cells are actually constantly kind of growing and shrinking in various parts because as membrane fuses with certain organelles and then gets pinched off to form little pockets or packages that we call transport vesicles. And those vesicles essentially are a way of moving larger molecules or molecules that maybe don't play nicely with other things, either because they're toxic or because they have particular properties, ways of moving them from one part of the cell to another, or bringing in molecules from outside the cell, or transporting things to the outside the cell. So membranes are basically membrane-bound organelles, and our vesicles are ways that our cells have of performing different kinds of jobs within the cell, and also shipping things either around inside the cell, using often those microtubules as little pathways or little roadways, basically, or shipping things outside the cell and moving them to another part of the body for multicellular organisms. So organelles basically do almost everything, right? They make protein, they transport molecules, they contain things that are potentially dangerous or hazardous. They're involved in recycling broken down molecules like enzymes that have been damaged, and breaking them down to their components, and then we can reuse the components for other things. So there's lots of different potential jobs, and we're going to talk about the jobs here of different kinds of membrane-bound organelles. But first, just a quick image of what difference basically between free ribosomes and bound ribosomes. Here we're actually looking at the surface under a scanning electron microscope of the rough endoplasmic reticulum, and so you can see where we have some bound ribosomes that are attached to the surface of the ER there, and then we have some other free ribosomes that are basically floating in the cytosol just outside the boundary of that particular organelle, whose main job is protein synthesis. So if we were to zoom in on one of the ribosomes, basically here's a diagram of what that would look like. So we have two different units here, the large and the small, and both of which are made of a combination of ribosomal RNA and protein. And then they lock together, and basically the messenger RNA is going to be going right in between the large and the small subunit when you have a messenger RNA molecule that needs to be translated into protein. So that's where the synthesis happens. Okay, so as we noted earlier, right, the ribosomes are not part of the endomembrane system because they are not organelles, right? They're not wrapped in membrane. Their job is very important, but it is not part of the ES. So as such, ES includes, as I mentioned, your plasma membrane to the cell, the nuclear envelope, and then all of our membrane-bound organelles, such as the endoplasmic reticulum, the Golgi that we're going to talk about here in just a minute, and lysosomes, peroxisomes, some other types of smaller organelles, and then all of our vesicles that we use for transport, and vacuoles, which are basically another membrane-bound space that's used for storage. Okay, so zooming in and talking a little bit more about the rough ER. As I mentioned, this is a membrane-bound organelle that's going to be studded with bound ribosomes, right? So the job of the ribosomes, of course, is making the protein, and the protein, as it's being synthesized, actually ends up inside the rough ER as it's being made, and therefore it can then be modified as necessary. There are actually additional steps that often need to happen with a protein. It's not complete just once it's been synthesized with the primary structure. It often will self-assemble and self-fold into secondary or tertiary structure but to get to its final functional form, it might actually need to be modified in additional ways, such as having other molecules or even minerals and things like iron and elements bound to it. It actually might need to be cut and cut up into pieces, or have little bits sort of shaved off of it. Essentially, you have to modify and sculpt that protein until its final form. And there are enzymes in the rough ER whose job it is to modify proteins to where they're in their final physical functional form, or they're in the form that's ready to be transported to another part of the cell or outside the cell where they will be where they're going to be used. Another major job of the rough endoplasmic reticulum is manufacturing membranes, right? So our membranes, of course, are it's mostly involved in rough ER is mostly involved in making the proteins that that are embedded within the endomembrane system, so different organelles or the plasma membrane. It's mostly involved in making the proteins there, but it also will make other membrane components. The smooth ER, which is usually found right next door to the rough ER, and we can see that actually on this little diagram here. So here's the rough ER with all of the ribosomes, and then the top left, you can see the smooth ER that is essentially lined up right next to the rough ER and almost touching it. And that's basically the organelle that is responsible for synthesizing the lipids that are part of our endomembrane system, right? So the phospholipids. And again, smooth ER, you'll notice it doesn't have any ribosomes. That's because its main job is not making proteins. Its main job is generally synthesizing lipids, but it also helps metabolize carbohydrates, so things that are more complicated, breaking them down into smaller pieces that our bodies can then use for fuel, our cells can use for fuel. It's also responsible for detoxifying some poisons, some metabolic poisons. So for example, folks who, your liver cells in particular, that's a big part of their job, is actually detoxifying metabolic poisons like alcohol, like ethanol, right, or other types of alcohol, act as metabolic poisons. So they, basically they damage molecules and they bind to things and make them not functional. So we need to get rid of that. So folks who drink a lot of alcohol, quite often their liver cells have their smooth ER of their liver cells is very enlarged because it's doing a lot of detoxification, and so it really needs to have more space. And that's kind of a sign that maybe got to cut back on that alcohol. But all of our cells have a smooth ER to help detoxify some metabolic poisons, and actually, oddly enough, store calcium. So again, calcium it's an important electrolyte actually, and it's really important for bone formation. It plays a key role in some other metabolic processes, and so our cells keep a supply of calcium on hand that's stored in the smooth ER. Okay, that brings me to the Golgi. This is actually, if I have a favorite organelle, this is among my favorites just because it's cool. And I've got a video that I'm going to show you guys in lecture actually on the cell when we get through the cytoskeleton. And we can actually watch the Golgi move. And it's very, very cool. So our Golgi apparatus is basically a network of membrane-bound organelles that are all in physical contact with each other. And if you had, right, we have the rough and smooth ER, right, or our membrane factories, our protein factories, our nuclear membrane, our nuclear nucleus, right, is our information storage center. If you want to think about, if you were a town, right, the Golgi would be our shipping and receiving center. So it's very much like the post office, right? or or in some cases, right, the UPS store, wherever you go to ship your packages. So the packages, right, are membrane-bound vesicles, and you can see how they're coming in on what's called the cis face. And just basically just same side. So you have things coming in from other organelles or coming in from the rough or smooth ER. And those membrane-bound vesicles then merge with the membrane of the Golgi on this end, and you can see that in the merging vesicles on the top part of this scanning electron micrograph of the Golgi in a cell. And they basically merge with the membrane and then dump their contents into the Golgi. And then the contents are, which are generally proteins, but it could be other stuff, are going to be modified and essentially tagged with molecules that say where they're supposed to go. So it's almost like how you have shipping labels on proteins, and then those proteins or other molecules get repackaged by the Golgi and shipped out into the cell to go wherever they're supposed to go. So their destination, may be other organelles, or their destination may be outside of the cell. And so it's going to get modified basically and repackaged. And then here's the transphase, and that's basically where you have membranes butting off to form vesicles, and then are going to ship those things elsewhere in the cell. So this is just a sort of a zoomed in look at the potential pathway of molecules that are going through the Golgi. So here we have a process known as exocytosis of a membrane, and we're going to talk about the difference between endocytosis and exocytosis when we talk about transport next week. But essentially you have molecules that were manufactured in the rough ER, and then basically you have a little membrane that's being pinched off to form a vesicle. That transport vesicle then will move along our cytoskeleton to the cis side of the Golgi, dump it, basically merge then with the membrane in a process called exocytosis, membrane of the Golgi, dump its contents inside, and then the contents you can see gets modified and then repackaged and may go elsewhere. So one of the destinations may be outside the cell, in which case that vesicle, transport vesicle, merges with the plasma membrane and dumps its contents outside the cell. And that again may be some, in contact with, for example, our bloodstream, and so you're dumping those chemicals into our bloodstream. You also may move molecules from the Golgi to other vesicles and other organelles like the lysosome or peroxisome if it's molecules that are damaged and need to be recycled or in some cases destroyed. Okay, so speaking of recycling things, I've got a little, this is a little, another kind of schematic drawing of a cell, right? And so here we've got the lumen just basically means fluid inside something. So in this case, it's the fluid inside the rough ER. And of course, you can see the little black ribosomes that are studying the surface. And then in this case, we have ribosomes are making a few different kinds of protein, right? So it's making maybe a protein that's got to go outside the cell. Those are the little red lines. And so once it's made, right, you butt off a vesicle. The vesicle merges with the Golgi. The Golgi repackages it and sends it outside to the outside world. And you have a different kind of protein that's also, again, getting repackaged by the Golgi and sent somewhere else, again, outside the cell. And the first one's little red lines actually became outside the cell, but part of the plasma membrane surface. Others are just being dumped outside. And then you have some that are basically being synthesized or being repackaged in the rough ER. And those actually end up probably enzymes that are supposed to function in the lysosome. So you have proteins being synthesized sent to the Golgi for repackaging, and then you get another vesicle that merges with the lysosome. So lysosomes are organelles that are essentially little pockets or packages of hydrolytic enzymes, right? And remembering, right, if you're going to form a covalent bond by condensation reaction, which is how we make a lot of our polymers, right? You take the water molecule out. Hydrolysis, or as actually a student said to me, hydrolysis, and which is not how you pronounce it usually, but it's a good way of remembering that you're using water to essentially cut the bond between two molecules. Essentially you're lysing it, right? So hydrolytic enzymes are enzymes whose job it is to break down macromolecules, to break down our polymers into their monomer components. And so here we've got enzymes that are being synthesized in the rough ER and then end up in the lysosome to do their job. And they do their job at a pH of about 5, which folks playing at home, right, can figure out how much more acidic that is than water. I won't do that here, but you certainly should be able to figure that out. So the job of basically the lysosome is essentially what I refer to as kind of the recycling center. It's taking all of our damaged molecules, right, or enzymes that are worn out, other things that are not working properly, like damaged sections of DNA, other kinds of molecules that just need to be cut apart, right? You bring in a chunk of protein from outside the cell, right, as part of digestion. You need to break that protein down into its amino acid components so that you can use those amino acids to build different kinds of proteins in our cells. So the lysosomes are very, very busy places because they're constantly receiving things that need to be broken down on a molecular level. And we refer to different types of enzymes. Most enzymes, their name ends in ASE. So if you see something that's like a protease, that ACE ending tells you, oh, this is an enzyme. And so it's going to be catalyzing a reaction. A protease is an enzyme that breaks down protein, right? Lipases are enzymes that break down lipids. Nucleases are enzymes that break down nucleic acids. And finally, I've just put polysaccharide aces because we have a number of different types of enzymes, and each enzyme focuses on a different polysaccharide. So you might have one that focuses on sucrose, which is actually only a disaccharide. It's a fairly simple sugar. And then you have some enzymes that break down much, much longer chains of starch, for example, or I'm blanking on the name of the enzyme that you guys are going to play around with in lab that actually does break down starch. And it starts with an A, and I can't think of it. But anyway, you'll learn about that in lab too. You're actually going to take some starch and you're going to break it down with this particular enzyme. So you have the names of the enzymes are often very specific to the complex sugar that they break down. We also have specific names for different types of proteases as well. We're going to be spending time talking about trypsin and pepsin and other specific enzymes like that. Okay, so just another little bit of a diagram involving a lysosome. So as I mentioned, lysosomes are going to be recycling damaged molecules as part of their job, but part of their job is actually also digesting food molecules that come in. Again, that might be a combination of carbohydrates and proteins, or it could be anything else, right, that's in our diet. We have to remember when we eat something, right? We're digesting generally things that are made up of other cells, and so those cells have nucleic acid in them. Those cells have lipids and other things that need to be broken down, and so we digest them outside of our bodies, right, with enzymes and acid and some other stuff. But eventually we get the particles small enough where we can move them through a vacuole, right? So, which is basically a type of vesicle. And so you have this molecule outside that we need to bring inside. So we wrap it in membrane, we pull it into the cell, and then it gets merged with a lysosome. And the lysosome, with the reduced pH and the enzymes, breaks that down into its component pieces, and then often just dumps the contents into the cytosol. So that those pieces can be then recycled and used to make protein or nucleic acid or whatever else that they can be reused for. So that's why I refer to our lysosomes as our cells recycling centers. Okay, peroxisomes. These things, frankly, are super, super cool. So a peroxisome is very much like if you think about your lysosome as a recycling center, your peroxisome is your hazardous waste disposal. This is where you bring, right, your old paint, your toxic chemicals that have been sitting around in your garage, stuff that's hazardous, that old car batteries, right, and you don't want the acid to get everywhere. So this is where our cells send the really nasty stuff that needs to be broken down. This is not like your run-of-the-mill metabolic poisons like alcohol. That goes to the smoothie R, right? These peroxisomes are getting rid of things that are really toxic, and you can't allow them to out into the cell because they will just cause tons of damage. And so what our peroxisomes do is to synthesize hydrogen peroxide inside, which hydrogen peroxide is a very corrosive chemical. And then you introduce that to whatever it is you want to break down, and the hydrogen peroxide basically dissolves it, well, not dissolves, but it essentially shreds it down to a form that is inert. It's not going to hurt anything. It breaks it down into a much less dangerous form. And then those chemicals again may be shipped elsewhere. The pieces that are left, what hasn't been completely destroyed by the hydrogen peroxide. What's also really cool about our peroxisomes is they will replicate themselves. So our DNA includes instructions on how to build all of our organelles in our cells, including the peroxisomes, but the peroxisomes can actually replicate themselves without DNA. So when it's kind of time to go through cell division, our peroxisomes will essentially just replicate themselves. And it's a process we don't really actually understand exactly how they do this. So I kind of love that there's still things about our own cells that we don't quite understand. It's a bit of a mystery, so figuring out exactly how peroxisomes replicate. I mean, they're basically little bubbles of membrane. So you have some of that self-synthesis process in the fact that they're just a lipid bilayer. And we know, for example, soap bubbles replicate, they just butt off of one another, but it's kind of interesting that we have that still going on inside of ourselves, and we don't quite understand exactly how they manage that. Okay, so I mentioned earlier that a vacuole is basically just a giant vesicle that doesn't go anywhere. That's essentially what it is. So it's basically just a membrane-bound pocket that are used to hold various different types of some substances. So food vacuoles, for example, you have phagocytosis or sometimes endocytosis. Essentially, cells wrap the food particle in membrane to form a vesicle that is enlarged and then merges with maybe the lysosome or some other digestive chemicals to break it down into its component pieces. Plant cells have a very large central vacuole that they use to store water. and sugar in the cell for energy that they can use at night when the sun is not up. They can burn that stored sugar and not have to move it from other parts of the plant, which when they run out, they have to move it from other parts of the plant where it's been stored. But individual cells, like, again, the plant cell in this diagram, which we know is a eukaryote cell, right? We've got a nuclear-bound, a membrane-bound nucleus here, right? We've got our Golgi, our endoplasmic reticulum, and then we also have mitochondria and chloroplasts that are in here. Those are those little circles that look like they're full of pancakes. That's a chloroplast, which we'll again talk about more here in just a second. But this, yeah, again, the central vacuole in plant cells actually helps support the plant cell by pushing on the cell wall and helping to make it rigid. So if it's full of water, which water being a very necessary component to photosynthesis, you absolutely have to have water. So that's a really important feature of plant cells in particular. But food vacuoles and other kinds of vacuoles are found in other kinds of eukaryotes as well. Okay, the mitochondria is the powerhouse of the cell. That's probably a lot of what people remember from high school biology. Our mitochondria really are, if you had different parts of the cell, and we talk about the different jobs, right? So the Golgi is the post office, and then we've got recycling centers and hazardous waste disposal centers, information center. Mitochondria would be the power plant. Now, that's not to say that cells without mitochondria cannot produce, basically cannot break down organic molecules into energy. They certainly can, right? We call them bacteria. They definitely function just fine. And in fact, what we think happened at some point was actually a bacteria started living inside a very, very early eukaryote cell. And we have some information about why we think that I'm going to cover here in just a couple of seconds, because we think that was the origin for the chloroplast in plant cells as well. But our mitochondria in particular are able to break down organic molecules like glucose and convert them to energy that's stored in ATP, which is a much smaller molecule, and you can use that, and you can move it around anywhere, and essentially spend it like cash energy. So our mitochondria basically have two membranes. There's an outer membrane that is contiguous with the membranes found in all of our other eukaryote cell components. And then we have the inner membrane that is basically a plasma membrane that has proteins that you find in bacteria and other prokaryotes. And then you get the folds in the inner membrane that allow for very small spaces in that membrane, and it's those little spaces that we're going to spend time talking about when we talk about cellular respiration. That's where all the action happens in terms of generating ATP from the energy that's stored in larger molecules. So our mitochondria are really fundamental components to how our cells release the energy that's stored in organic molecules. So the initial breakdown starts in the cytosol, but eventually things end up in the mitochondria, and that's where really all the action happens in terms of releasing that energy so that our cells can then use to build protein or copy DNA or doing anything else, moving things around. Basically have the cash to spend on other kinds of processes that require energy and that don't release it. I'm trying to think if there's anything else that I wanted to say about mitochondria. Oh, you also notice, right, our mitochondria have their own ribosomes. Interesting, right? And they're free ribosomes. They're just floating around. So the inside of a mitochondrium really does look like the inside of a bacterial cell. They even have their own circular DNA inside the mitochondria. I know, super, super cool. So we're going to spend a lot of time talking about mitochondria, like I said, when we get to cellular respiration. Okay, and in a very similar fashion, we're going to spend a lot of time talking about the structure and the process of structure of chloroplasts and the process of photosynthesis later on in the course after the first exam. But again, to know is also we do believe that chloroplasts started their lives as a free-living bacteria. They also have their own DNA, their own ribosomes, as we can see on the diagram here, and more folded membrane. In this case, membrane folds that look very much like little circular pancakes that we call thylakoids. And that's basically the site of the action in your chloroplast, in this case actually harvesting energy from sunlight and converting it to sugar. So basically the opposite of what happens in the mitochondria is what's happening in our chloroplast. And so again, we refer to them as chloroplasts. We have other versions of this that occur in other eukaryote cells that we refer to as plastids. And notice that both the mitochondria and the chloroplast, neither one of those are considered part of the endomembrane system. Not that they are wrapped in membrane, and it's eukaryote cell membrane, but they're not considered part of the ES because their origin is believed to be separate. We don't have DNA in our nucleus that codes for mitochondria or chloroplasts. So if you're a plant cell. So if we remove those, and folks have done experiments where they have used micropipettes to remove mitochondria from a cell or remove a chloroplast from the cell cannot make more of those. It doesn't have the directions in the genome to build those, which is rather interesting. So as I mentioned, we do have reason to believe that our mitochondria, and if you're a plant cell, chloroplasts, were once free-living bacteria that were essentially phagocytized, wrapped in membrane, and brought into a host eukaryote cell, and for whatever reason they were not digested. Right? Normally they would be subjected to digestive enzymes and and be done with it, broken down to its component pieces and recycled. But for whatever reason, our mitochondria and the chloroplasts in plant cells, that didn't happen. In particular, our chloroplasts are almost identical to living cyanobacteria, actually, down to the folded membranes that we call thylakoids. So they have a double membrane, right? The inner membrane, as I mentioned, is a prokaryote membrane with prokaryote proteins, and the outer one is a eukaryote membrane with eukaryote proteins. They have their own ribosomes. Again, they have their own DNA, and their DNA is circular. It's not linear like eukaryotes, right? So they have their own circular DNA, and they replicate themselves independently from the rest of the cell. So like I said, our cells, if we lose our mitochondria, we cannot make more. But prior to cell division, our cells tell the mitochondria, hey, we're getting ready to divide. You might want to think about copying yourself. And so the mitochondria actually do replicate their own DNA. They divide separately. They replicate themselves in a process that's almost identical to how bacteria replicate themselves, because bacteria do not use mitosis. They don't have the cytoskeleton components to do that. So they use a process that's called binary fission. If anyone just sort of they basically pull themselves apart until they butt off another cell. And our mitochondria replicate the same way, and so do chloroplasts in plant cells. And so they will replicate themselves prior to cell division.